You may be surprised that I'm wearing a tie today. Indeed, bioinformaticians are hardly ever dressed like this when they give talks. However, 60 years ago, James Watson and George Gamow formed the RNA tie club. The goal of this club was to decode the genetic code. There were 20 members in the club, one for each amino acids, and the mission was to figure out which codons code this particular amino acids. The members of the club met twice a year, smoked cigars, and drink wine. And moreover, they understood the importance of mathematics in biology, so one of these 20 members was a famous mathematician, Nick Metropolis, who actually was one of the founding fathers of randomized algorithm. However, with all this firepower, there were 8 Nobel laureates in the club. They have not decoded the genetic code. The honor of decoding the genetic code went to Marshall Nirenberg, who did it in 1961. Today, we will talk about arguably a more difficult task, decoding the non-ribosomal code. I'm sure that 99% of students in this class, before the class started, thought that the only way to assemble amino acids into a protein or a peptide was to have a ribosome and to use RNA as a template. However, as we learned in the antibiotics lesson, there is another highly unusual mechanism for producing peptides and assembling amino acids into peptide chain. It's called non-ribosomal peptide synthesis. And Mohamed Marahel, 15 years ago, set on the goal to decode the non-ribosomal code. An extremely difficult task. Non-ribosomal peptide synthesis is performed by a giant protein called non-ribosomal peptide synthetase, or NRP synthetase. And the task that Marahel was facing was this one. NRP synthetase in Bacillus brevis, as we learned, code 10 amino acid peptide tyrosine. But what particular amino acids in NRP synthetase are responsible for synthesizing, let's say, valine in tyrosine? Non-ribosomal synthetase in Streptomyces rhozosporus codes for antibiotics of the last resort called daptomycin. But at the time when Marahel started, it was absolutely unclear what particular segment in NRP synthetase in Streptomyces rhozosporus codes for daptomycin. However, Marahel knew something about non-ribosomal peptide synthesis. It is performed, as I mentioned, by a giant molecular assembly line, NRP synthetase. And for every amino acid being synthesized, there is a segment in NRP synthetase, roughly 600 amino acid long, called adenylation domain. So this adenylation domain synthesizes one amino acid at a time, and since tyrosine is 10 amino acids long, there must be 10 adenylation domains in NRP synthetase in this case. So what are particular amino acids, or what is the non-ribosomal code that decides what amino acid to synthesize? Well, Marahel started by taking a look at some adenylation domain, and he argued that despite, let's say, these 3 adenylation domains code for different amino acid, there must be something similar in them because they all try to accomplish the same goal: add one amino acid to the growing peptide chain. He looked at the amino acids in these 3 adenylation domains and found very little similarity. There is hardly any similarity here, only 3 conserved columns, which is statistically insignificant. However, if you just start shifting some of the sequences, like I just did, you see 11 conserved columns. And if you continue doing this, suddenly you reveal 19 conserved columns. This is statistically very significant, which reveals that these adenylation domains are indeed similar. We have just constructed the conserved core of adenylation domain. The next line of attack for Marachel was to figure out which positions in these adenylation domains are actually responsible for coding different amino acids, which of them are different. And he succeeded by finding 8 blue positions that are actually responsible for specificity of amino acid synthesis. As a result, Marachel was able to crack non-ribosomal code, and it turned out to be extremely complex. 8 amino acids located in different parts of adenylation domain code a single amino acid as shown on this slide. But if we were not able, if we would not move and shift some amino acids in some sequences compared to other, we would never be able to find conserved blue column. So it now looks like a card trick, a magic trick that Marachel performed. How in the world he was able to figure out how to align sequences in such a way that he was able to reveal the conserved core. Today, we will learn how biologists compare biological sequences, but before we move there, I will tell you about one more biological success story, the story of hunt for the cystic fibrosis gene. We now move to 1980s, when biologists were trying to find the cystic fibrosis gene. Cystic fibrosis is an often fatal disease of the respiratory system that produces an excessive amount of mucus, a slimy material that is secreted into fluids such as saliva. Cystic fibrosis is a rather frequent genetic disease. In the United States, 1 in 25 people carries a faulty cystic fibrosis gene. It doesn't mean that they have cystic fibrosis, but if 2 people with faulty cystic fibrosis genes have a child, there is a 25% chance that the child will have cystic fibrosis. When in the late 1980s, biologists narrowed the search for the cystic fibrosis genes to small regions on chromosome 7, the question arises which particular gene in this region is responsible for the cystic fibrosis. And at that time, genetic mapping techniques were limited, so they could not narrow the search further. As a result, they started to look at these genes and decided to compare each of the candidate genes in these narrow regions to all known genes at that time. And one of these comparisons revealed that one of these candidate genes on chromosome 7 is similar to an ATP binding protein that acts as transport channel responsible for secretion. Remember, cystic fibrosis is related to secretion, and ATP binding proteins are also related to secretion. That was an eureka moment, and biologists conjectured that this specific candidate genes, thanks to the similarity search, is actually the gene responsible for the cystic fibrosis. They call this gene cystic fibrosis transmembrane conductance regulation, or CFTR. And the CFTR protein controls the flow of ions in and out of cells inside the lungs. If a mutation happens inside the CFTR protein, that the flow of ions is disrupted, and it leads to cystic fibrosis. In fact, it turned out that just 3 nucleotide deletions in this gene disrupt ion channels and causes cystic fibrosis. And now the time has come for us to look at what algorithms biologists use to align adenylation domains or to compare various candidate genes in the case of search for cystic fibrosis.